Hello everyone, my name is R.Vinodhini, Assistant Professor in the Department of Biomedical Engineering, School of Bioengineering, Bharat Institute of Higher Education and Research. I am handling the subject Medical Informatics. Today we are going to discuss about the topic Bioinformatics. So first, what is meant by Bioinformatics? It is the combination of two fields, biology and information technology. So it is the combination of biology and information technology. It is a recently developed science using information to understand the biological phenomena. So with the use of an information technology, we can able to get an information or understand the biological phenomena. It involves the computational tools and also various methods which is used to manage, analyze, and manipulate volumes of a biological data. It may also be regarded as a part of a computational biology. Later, it is concerned with the application of quantitative analytical techniques in modeling and also solving problems in a biological system. So, it is an application of a quantitative analytical techniques in modeling and solving problems in the biological system. It is an interdisciplinary approach requiring an advanced knowledge of computer, mathematics, and statistical methods for understanding of the biological phenomena at the molecular level. And let's see about the history of bioinformatics. It was first introduced in the year 1990. It dealt with the management and analysis of the data pertaining to DNA, RNA, and also protein sequences. It includes many other types of biological data. Most important ones are like gene expression profiles and also protein structure. So these are all the two important ones which we can able to identify using this informatic technology. So these are all the two important biological data so you can able to identify using the information technology. So those are gene expression profile and another one is protein structure and protein interaction, then microarrays like DNA chips and the functional analysis of molecular biomolecules, then drug designing. So all those things can be able to identify using an information technology. And this it is a broad coverage of an bioinformatics. We can able we are having a functional genome, structural genome, comparative genome, and DNA microarray medical informatics. Okay, so functional genomic means it is an identification of genes and their respective functions. It is what we call it as functional genome. So with the use of this bioinformatics, what are all the things we can able to done? These are all the things like a functional genomic, like able to identify the genes and also its functions. Then structural genomic, we can able to predict the relation of the functions of the protein. So we are not clear about the protein function, but we can able to predict using this bioinformatic tool. And then comparative genomic, here we can understand the genomes of different species of an organism. So there are variety of species were there and it has a variety of genomes. So we can able to understand the genomes of a different species. Then DNA microarrays. So here it is to designed to measure the level of gene expression in different tissues and in a various stages of development and in different diseases. So designed to measure this DNA microarrays are designed to measure the level of gene expression in different tissues and also various stages of development and in different diseases. And then medical informatics. It involves the management of biomedical data with the specific references to biomolecules, in vitro assay, and also clinical trials. So these are all the basic concept we can able to get using this bioinformatics. And components of bioinformatics. We are having about three components. So first thing is creation of databases. Here, it involves the organizing, storage, and also management of the biological data sets. So, first point is that involves the organizing, storage, and management of biological data sets. And the second point is databases are easily accessible, and also we can able to submit the new entries. Then protein sequences data bank for the molecular structures. So this is the first component, creation of a database. So with this use, if we create the database, what we can able to do, we can able to manage all the biological data sets. We can store and also organize the different biological data, and also able to submit the new entries, and the protein sequences data bank were there. So there are different kinds of molecular structures, different kinds of genomes were present, right? So each have a unique data bank. So if a particular data protein sequence is identified, it is stored in the same bank. So this is the concept here in creation of database. And the second point is second component is development of algorithms and statistics. So here it involves the development of tools and resources to determine the relationship among the members of large data sets. So it is involves the development of tools and resources to determine the relationship among the members of large data sets, and also able to compare the protein sequences data with the already existing protein sequences. If you identify the new protein, we are not that much clear about that protein sequence, right? So in such case we can compare the new protein sequence to the already existing protein sequences. So this is also the important thing. So with the use with this development of algorithm and statistics, we can able to compare the old sorry, compare the new protein sequence with the older one. So that is the concept here. And the third component is analysis of data interpretation. So here what they are trying to tell is includes DNA, RNA and protein sequences, protein structures, gene expression profiles and biochemical pathways. All we can able to analyze here. So analysis of data interpretation, it includes DNA, RNA, protein sequences, protein structure, gene expression profiles and biochemical pathways. So this is what about the components of bioinformatics. Then relationship between the bioinformatics and the internet. So it is an international computer network which involves a group of computers which can be communicate and also exchange the data between the users. So once the user ID it has been created, means we can able to share the data anywhere in the world. So for that we need internet, right? So it is much more important for sharing the data, and also to have a, data set, biological data sets. So it involves a group of computers that can communicate and also exchange the data between the users. So the IP, Internet Protocol, is there, right? It determines how the packet of informations are addressed and routed over the network. So this Internet Protocol plays an important role here. And to access the internet, the computer must have the correct hardware, appropriate software, and also permission for access to the internet. So for sharing the data or to store the data, so these are all the main important thing here. It is to access the internet, and also computer must have a correct hardware and appropriate software, and also like permissions for access to the networks. So this is the main thing. And application of bioinformatics. So where these bioinformatics were applied, like a sequence mapping of biomolecules, like DNA, RNA, and protein sequences, we have to map, right? So in such cases, it is much more needed thing. And identification of nucleotide sequence of functional gene. Identification of nucleotide, it is important, and then finding of sites that can be cut by restriction enzymes. The third point is finding of sites that can be cut by restriction enzymes, and also prediction of functional gene products. So last point is the prediction of um, functional genome, and to trace the evolutionary trees of the gene, and for the prediction of 3D structures of the protein. So for the prediction of 3D structure of the protein also, it is much more used. And the molecular modeling of the biomolecules, then designing of drugs for medical treatment, and also handling of vast biological data with which is otherwise not possible. If you if it is not in this bioinformatics field, we cannot able to handle that much of biological data, right? It cannot be in an hard copy, or we cannot able to entry the data. So this informatics is much more needed thing. So handling of a vast biological data, which is otherwise not possible at all with the common entry. and development of model for the functioning various cells, tissues, and also organs. Then we'll move on to the software. So if you go for the software tools for bioinformatics ranges from simple command line tools to more complex graphical programs and standalone web services available from various informatic companies or public institutions. So software tools for bioinformatics ranges from a simple command line tool to more complex graphical programs and standalone web services also available from various bioinformatic companies or a public institution. So if the person is having a much interest on this bioinformatics, they can able to create a new softwares, new programming tools to identify the data, biological data sets. And the computational biology tool best known among the biologists is probably BLAST. So BLAST is just the software which is used, which is an best known among all the biologists. Okay, why why? Because it is having a algorithm for determining the similarity of arbitrary sequence against the other sequence. So we can able to compare the sequences of DNA, RNA, other protein. Okay, so it is much needed software here. So it is possibly from curated databases of protein or a DNA sequences. So already they will be having a set of identified databases. So whenever they need to identify the new protein sequence or a DNA sequence, they will be able to compare these two data. So it is the most common used software. So this BLAST, it is one of the number of generally available programs for doing sequence alignments. So commonly for what purpose it has been used is for the sequence alignment. This BLAST is used, and the NCBI provides a popular web-based implement that searches the databases. I'll tell you about the explanation of the NCBI and also the BLAST. So BLAST is one of the commonly known software for doing a sequence alignment. For doing the sequence alignment, this BLAST is used. So it is provided by NCBI, which is a popular web-based implementation that searches the databases. Yeah, BLAST. So BLAST, it is a basic local alignment search tool. BLAST, it is a basic local alignment search tool. It is an algorithm for comparing a biological sequences informations like amino acid sequences of different proteins or a nucleotides of an DNA sequences. So like different proteins like amino acids and different DNA sequences like nucleotides can be able to compare. So it is having a set of algorithm to compare the newer data with the already existing one. So BLAST expansion is the basic local alignment search tool. It is a software created by NCBI. So this BLAST is used to identify library sequences that resembles the query sequences. BLAST is used to identify library sequences that resembles the query sequences. It is a tool used for alignment of the sequences. So I told you before itself, BLAST, it is the tool for an alignment of sequences. BLAST was originally developed by NCBA, National Center for Biotechnology Information. So BLAST was developed by National Center for Biotechnology Information. So they only created this tool software so that all sequences of protein, DNA, can be able to compare here, and all the data are stored. So they will be having a user ID here. Only those persons can able to get into the software, that web page. So others cannot able to just like that go and search for it. So that is the main thing here. It has been fully provided with the like fully provided with care. So this NCBI site provides a binary files for the following operating systems. So what are all the operating systems and platforms this NCBI having, like Unix, Linux, Mac, MS Windows, and IBM AX 5.1. So all these will be used for the comparison. So it's like a kind of library only. So in a library, they will be having a arrangement, right? Like alphabetical arrangement or a binary arrangement. They will be having a set of books. The same way, here the set of biological data has been stored one by one. That is the concept here. It is same like a library in a virtually it has been placed here. E-library, like we can tell it as e-library. So if you put any sequence name or a protein name, we'll be getting a combination of all those data. And uses of blast, what is the main uses of blast is, it can be used for several purposes. It includes like the sequence of interest, identifying the species, identifying the species, it is the main use here. So with the use of the blast you can possibly collect, possibly identify a species and also find out the homologous species. So if you found a new species, we can able to identify what with the help of its protein set or a DNA set, we can able to identify which kind of species is that, and also we can store the uh, store the species in the in its particular family. So that is the concept here. So with the use of this blast, you can possibly correct, possibly almost we can able to correctly identify a species and also like a homologous species. So if this belongs to the particular family, it can be able to easily identified. It is this can be useful for example, when you were working with a DNA sequence from an unknown species, it can be correctly locate the class or a family which the that species belongs to. So if you get a, if you have a protein or a DNA sequence, we can able to easily identify the new species family. So that is the main use here. So main use of this blast is to identify the species family and also locating domains. When working with the protein sequence, you can input it into a blast to locate known domains within. So if it is in a if we are working within a DNA family, we can able to easily identify its family, what species it belongs to, that can able to identify. Whereas if you have a protein sequences, it can be input it into the blast to locate the known domain within. So if you know the protein sequences, it can be able to locate its domain. And also comparison, and this third thing is comparison. When working with the genes, blast can locate the common genes in two related species and can be used to map annotation from one organism to another. So DNA sequence, if you're working with the DNA sequence, we can able to find out its family. If you work with an if you work with an protein sequence, we can able to know its domain. And if you work with the genes, we can able to locate a common genes in two related species. If the two species it is having a same gene, we can able to identify from which family it is belongs to. So those comparisons also we can use with the use of this blast. Look at this. This is the web page or a software to identify the different kinds of protein, genes, DNAs, etc. So if you see, they'll be having a nucleotide, protein, genomes, translated, special meta. So all those things will be there. If you go for the search domain, if you enter the protein sequence, you can able to get all the related all the related protein gene combination and genomes, like it is belongs to the human genre or a mouse genre or a rat, so all those families also we can able to identify, not only for the humans and also for the species like microorganisms, also we can able to identify. And translates, like translated query versus protein databases. So if you know the only the translated query, we can be able to search with the protein database. So that kind of things are also able to identify with the use of this blast software. So it was created by NCBI. And see, download where data. So if you want to save the data, you can able to download it also. So how to use this software, for that they'll be having a help desk here. If you want to find out any new combinations, uh, they'll be having a help desk there. You can search and type, get a idea or get a knowledge of it. So this is the web page for a blast. Then FASTA. So if you go for the FASTA, it is a DNA and protein sequence alignment software package. So DNA and protein sequence alignment software package. So if I told first we have seen about the blast, right? So if there we can able to identify the DNA alignment, RNA alignment, protein sequences, everything. Here this from this FASTA we can able to identify the DNA and protein sequences alignment. So it is also a kind of software only. So this FASTA package contains the programs for protein, like a protein, DNA, protein, DNA, protein, translated DNA, and ordered or unordered peptide searches. So all various combinations we can able to identify with this use of FASTA, but only these two things, DNA and the protein sequences only. So we can able to found out protein sequences, or the DNA sequence, protein and the translated DNA. So those comparisons we can able to get using this FASTA. And last, we'll came to the conclusion. Here, in this conclusion part, we are telling about how this bioinformatics is widely used in the sphere, in the field of biology. So, with the use of information technology, this biological data has been saved, stored, and we can able to create a new ideas with this help of the information technology. So this bioinformatics, it is a combination of two main fields like biology and information technology. So with the combination of these two, we can able to identify the vast things in the biology field. So the recent enormous increase in biology data, it has made it necessary to use the computer information technology to collect, organize, maintain, access, and analyze the biological data. It is quite easier and like a knowledgeable way for identifying the biological data. So all the data can be easily stored, and we can able to share the data whenever we needed with the use of the internet. These are all the importance advantages of this bioinformatics. And the main thing we need is the computer. We have to check for the computer software and hardware also. It has to be properly fixed. So computer speed, memory, exchange of information over the internet, it has greatly facilitated bioinformatics. Then the bioinformatics tools available over the internet and are which are accessible, well developed, fairly comprehensive, and relatively easy to use. So these softwares are easy to use to the biologist, and also it cannot be easily theft. Why? Because it has a it has a security private privacy is there, so it cannot be easily theft. The data will be stored properly, and only the authorized person can able to exchange the data. Why? Because if the unwanted things were sent to the wrong person, means it will be like a problem to the persons, right? So all the stored data will be uh, safely secured there. So this is the main thing in this bioinformatics. So here the main use here is to collect, organize, maintain, and also access and analyze the biological data. So and the second main important thing is that computers, so its speed, memory, and the exchange of the information over the internet. So these are all it is quite interesting and very much useful for the biologists to transfer these data. And what are the other things like bioinformatics tools available over the internet are accessible, well developed, fairly comprehensive, and relatively it is easy to use. So this is the main thing in uh, bioinformatics. So that's all about bioinformatics. So we have seen about the introduction part, what is meant by bioinformatics, and then we have seen the applications. So where are over which field it has been applied, and then components we have seen. So there are about three components there. So those things we have seen, and about the two softwares. Mainly in this bioinformatics, software plays an important role. So BLAST we have seen, and the FASTA. So BLAST, we can it is developed by NCBI, where we can save, search, or compare the data using the previously set data. So DNA, RNA, and protein sequences can be able to identify in the BLAST, and by FASTA, DNA sequence, protein sequences, and protein to translated DNA sequences, all those can be able to identify. So this is the difference between BLAST and the FASTA software. So that's all about bioinformatics.